You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that had, has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you. Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast series. I have Emeran Mayer. He's a professor at UCLA, professor of physiology in the Microbiome Center. He's got over 300 publications, 320 peer-reviewed articles, so not too shabby. We're going to be talking about his current research on the role of gut microbiota in modulating brain-gut interactions and their role in emotion regulation, chronic visceral pain, and obesity. So this will be great. Emeran, thank you for coming.
It's a pleasure to be on the podcast.
Yeah. I guess just attacking it head on, how was it first established that there was the possibility or the likelihood of the reality that our gut microbiota affect things like emotion and obesity and other physiological functions?
Well, I would say, the history, if you go in the pre-scientific history of that, I mean, this has been a fairly popular topic in the 18th century. People went through these hycolonics. There's some a French king who actually went through this every day to improve his well-being, and I think it even goes back to the Egyptians who believed that they didn't know that these are microbes or microorganisms, but that there's something going on in the gut that affects well-being, both physical and emotional well-being. In terms of the scientific approach, there were in the last century, Metchnikoff, a person that actually won the Nobel Prize, who promoted the regular intake of probiotics, of microbes with a potential beneficial effect on health, as a important modulator of well-being. Scientifically, this started actually much more recently when the first animal experiments came out where people used germ-free animals and found that either that transplanting, for example, fecal material from mice that had a particular genetically determined emotional behavior, you could transfer that behavioral trait into the recipients, into the germ-free recipients, just by transferring the fecal material. And there have been a series of similar experiments since then in mice treated with broad-spectrum antibiotics, various other manipulations that clearly have established that at least in mice, in the mouse model, there is some kind of a relationship between gut microbial composition and the metabolites that they produce and behavior.
I've heard things like 90% of all serotonin is made in the gut. The gut's not defined. I guess it would be the proximal colon. I've heard that B vitamins are made there, and that thyroid hormone T4 is transmuted into T3 there. But I haven't found specific places in the literature where they're saying, who, meaning what species or multiple species are doing it. Have you seen any such thing that I've heard these things and then corroborated them in the literature?
Yeah, I mean, sticking with the serotonin story, I mean, that is still a puzzle because we don't know why, the great majority of serotonin is not located in the brain, where we, treat patients with depression with serotonin reuptake inhibitors, but it's in the gut, and we do know some things. It affects, it's important for peristaltic reflex and for secretion of fluids into the gut. But it's still surprising why we would have this massive amounts there. Now, there's recent experiments from the laboratory of Elaine Schall at UCLA. She's a collaborator. And so she has first demonstrated that certain microbes, certain Clostridia, play an important role in stimulating the synthesis of serotonin from tryptophan in these cells that contain, that are these storage sites, the so-called enterochromaffin cells, and that at least in the mouse, a large percentage of the synthesis is dependent on these Clostridialis, these taxa of microbes. And the way they do it, she also showed that, is by using short-chain secondary bile acids and short-chain fatty acids to stimulate this production. So one important mechanism is obviously that they help in converting the amino acid tryptophan that comes with the diet into serotonin in these cells. And then she's shown in a more recent study, which is equally fascinating, that similar taxa of microbes also have a molecule that is very similar to the serotonin transporter, by which human cells, nerve cells, and platelets reuptake the serotonin that's secreted. So the microbes have a very similar molecule that can take up serotonin that obviously must be in the gut lumen, because that's where the microbes live. And so the current idea is that there is an intricate relationship between certain microbes stimulating serotonin production. Some of that serotonin is released onto vagal nerve endings and goes to the brain. Other portions are released onto the enteric nervous system neurons to regulate peristalsis. And another portion is secreted back into the lumen of the gut, where it's taken up by microbes, sort of completing this circle. What effect the serotonin in microbes has on the microbial behavior, gene expression, is currently under investigation. So an unfolding story that I think we don't know the final outcome yet, but I think it certainly points to an important role of specific taxa of microbes in tryptophan and serotonin synthesis and metabolism.
Something you said sticks out at me. When you said that serotonin may directly stimulate the vagus nerve itself. I mean, it wouldn't certainly travel along the vagus nerve. I thought it would have to travel through the bloodstream and then maybe go through the blood-brain barrier to go into the brain. But are you saying that there's a possibility that the brain can be directly stimulated and modulated itself using the vagus nerve in the gut?
Yeah, and that's another puzzle about this whole serotonin story. So you would think, if there's all this serotonin stored in these cells, they would just release it into the bloodstream, and then some of it would get to the brain and, modulate. I mean, obviously serotonin in the brain can modulate most of the basic functions, from emotions to appetite to pain modulation. But that's not the case, because the serotonin that's released into the bloodstream is immediately taken up by platelets, and then the platelets carry to distant sites in the body, to our knowledge, current knowledge, not to the brain. So there's got to be another mechanism. And also relatively recent research in the last couple of years has shown that vagal nerve endings, so sensory vagal nerve endings, form synapse-like connections with the serotonin-containing enterochromaffin cells. So they're not just in close proximity, but they're tightly connected in a synaptic way with these cells. So if a serotonin-containing cell is stimulated from the gut lumen, from microbes, it will release serotonin into this synaptic gap, and it will activate directly vagal afferents that then carry it into the brainstem and from there to other brain centers. Again, what role this particular type of this component of serotonin plays in our emotions, as compared to, for example, the serotonin that's released from cells in the brain, in the brainstem, is currently not known. But it's certainly, we know the mechanism is there, and obviously it must have some role in the overall regulation of our behavior.
But that's kind of a crazy implication, because I may be getting this wrong, but that means that essentially the brain has a part of it, again, the vagus nerve that's, sitting in or around the gut, that can be stimulated in such a way as to directly modulate brain activity, as if you were in the brain modulating the activity. It's like a tendril of the brain hanging down into the stomach that can be affected by the stomach directly.
Yeah, and that's
exactly the case. That's crazy.
Yeah, well, I mean, that's exactly the image I think one should have. It's almost like the eye, which is connected through the optic nerve to the brain. So you could say you have these gut cells located in the periphery in a place where they're exposed to all kinds of messy things and all kinds of metabolites that there's a direct access to the brain. Now this access cannot be something that would affect your behavior on a minute by minute basis, because otherwise our brain couldn't really function. Our emotions would constantly switch depending on what you eat. That's another puzzle. I personally think that these are tonic influences that provide a background serotonergic tone to the brain, rather than phasic, second to second or minute to minute influences. But again, to what component that what role this plays compared to the serotonin synthesized in specialized neurons, we can't currently answer that question.
Well, this is interesting. This is forming in my mind could you look at the gut as literally a sensory organ of the brain? It's a slow informing eye or ear, or it's literally like a sense, a sixth. Again, I'm not going to be metaphysical about it, but it literally is. Since we have five, it literally is the sixth sense. It may not have a tremendous impact, or it may have a slow impact, or like you said, a background impact, but I don't know, that's the feeling I'm getting from what you're saying, that's the possibility. What's your thought on that?
Yeah, I mean, I couldn't agree with you more. So we talked about serotonin. I mean, there's other what we call enteroendocrine cells in the gut. If you put all these cells together, it would be our biggest endocrine hormone producing organ. And so one well-known example are these hormones that are essential for creating a sense of satiety. There's NPY, and probably one of the most well-known of this group of satiety hormones. They are located in cells just like the serotonin cells. They're cousins, they're comparable, but have a different function. So they're being released again from microbial products, short-chain fatty acids. The receptors on these cells match the signals that the microbes produce. Serotonin, not serotonin in this case, but short-chain fatty acids, secondary bile acids, and these cells then release these hormones. Some of them, similar to what I told you about the enterochromaffin cells, get onto these by synaptic transmission onto vagal sensory nerves, so they have the same vagal pathway to the brain. Others get into the bloodstream. And in this case, they can reach the hypothalamus through the bloodstream and are the main mechanisms to create the sensation of satiety after a meal. There's a lot of research on this, how these mechanisms become compromised, so satiety doesn't set in and it leads to overeating. In mouse models, that a high-fat diet would basically downregulate the sensitivity of these cells, so mice would not get sufficient satiety and keep eating. And there's several of these cells. There's also in the stomach cells that contain this hormone ghrelin. It's the only gastrointestinal hormone that actually stimulates appetite and food intake. And again, there is influences from microbes on this cell as well. So what you said is absolutely right. I mean, the gut is probably the biggest sensory organ that we have. It's the surface area. If you spread it out, all these villi and microvilli, if you would spread all these out onto a flat surface, it would be the size of a basketball court. And that surface is studded with all these sensors. And make it even more complicated, the sensors, these different chemicals that they release interact with each other. And so you have I would say, in our body, certainly the most sophisticated sensory organ that informs the brain about many aspects of, food intake, but also, as we talked about earlier, about emotional context, emotional state. And I would say we're just, even though the science is already pretty exciting, I think we're just at the beginning really of understanding the entire system. And we'll probably, it's intense effort, research effort goes into this. But I would say within 10 years, we'll have a pretty complete understanding, and which obviously will open up possibilities for therapeutic interventions, on a large scale.
Well, since obviously food is super important, otherwise, we don't live. And food, so we eat food, maybe not in the mouth or the esophagus, but once it gets to the gut, once it gets to the stomach and then the intestines, now it's either going to be integrated into our body or not. And it's from the outside world. So it makes sense that the stomach and the gut, it's a critical place. It's an intake source or location of where we're getting our nutrients. So it really needs to be monitored, and it makes sense that this is a sixth sense. But I wonder if you were to rank the senses, hearing, sight, touch, etc. This seems to be the most microbe mediated or microbe controlled. Like eyesight, I do know from the literature there is a microbiome of the eye, but the main functioning and control mechanism in the eye appears to be the somatic cells of the eye themselves, as with other senses in our body. But this sense, if we're to call it a sense, appears to be, again, the most microbe-mediated and or controlled of all the senses.
No, absolutely. I mean, every surface in our body has a microbial community, ecosystem. So the skin, the urinary system, pretty much. I mean, our entire surface is covered with microbes, and they're all specialized, and they produce, particular molecules or mediators that have some influence. But clearly, in terms of influencing such vital things like ingestive behavior, essential for survival, pain perception, emotional behavior, I don't think there's any system, and we can say this today already, with our limited knowledge. I don't think there's any system that is that closely connected to the microbial world. And it does make a lot of sense if you go back in evolution. The first marine animals is called the hydra, these tiny animals, which were essentially floating digestive tracts. They were surrounded by a nerve net that regulated the peristalsis of that floating gut. And when I say floating gut, so basically this tube had an opening on one side where it sucked in water from the outside, and it had at the opposite end would expel undigested or unabsorbed components, or what it took up. That sort of this integrated relationship that developed early on between microorganisms living in the oceans, and then at some point finding out that it's actually convenient to live inside that tube, because these microbes got free transport, protection, unlimited supply of nutrients. That symbiosis of microbes both with the digestive system, so the tube that they lived in, but also with the precursor of our nervous system, these nerve nets, that has persisted throughout evolution. Virtually every creature today, from insects to mammals, has that arrangement, that there's microbes living inside the gut, and these microbes can communicate with the nervous system, both the what's called the enteric nervous system, which was the one that was there first, so it was really our first brain, but also with our central nervous system, which is really our second brain. So it makes a lot of sense that we have this unique role of the gut with its sensors, compared to all the other areas of the body that also has microbes living on them.
And when again, the word gut is thrown around a lot, and I realized, like, where is the major innervation? Where is the connection to the vagus nerve? Is it in the colon? Is it in the small intestine? Is it spread throughout the whole digestive system? Like, if you look at it, where is the majority of the connection happening?
It's it varies with species. I think all species share the same innervation from the stomach to the sort of to the mid colon, or towards the last third of the colon. The last third of the colon in humans apparently is not as densely innervated, or not innervated by the vagus at all. I think in some mice that innervation exists, but you could say that in humans, the entire GI tract is innervated all the way down to the last third of the colon. And that's important also in terms of the microbial communication with the vagus nerve, because even though the highest numbers of microbes are in the colon, there's lots of them starting really in the beginning of the intestine, from the proximal jejunum down into the terminal ileum, much lower, magnitudes of order of magnitudes lower than in the colon, but still a very large number. So I think a lot of the mechanisms that erroneously are blamed or associated with the colon really happen in the small intestine, because you don't need billions of microbes to signal, for example, to these enterochromaffin cells or enteroendocrine cells. Even 100,000 would be sufficient to do that signaling. So I would say, and this is just starting with the development of sensors and smart pills that go down the entire digestive tract and sample at different locations of the stomach and the gut. And people have shown that there's distinct microbial communities at all these levels, from the stomach all the way down, to the mid or end of the colon.
Well, one thing I've found informative, and I don't know if this will directly translate, but I think it will. I've spoken to people about the oral microbiome, and there's multiple niches. There's, on the tooth, above the gum, below the gum, the top of the tongue, the bottom of the tongue, the back of the throat, etc. So just in that one area, probably according to cell type, there's a microbial attachment to each, a different one. And then I spoke to a researcher named Florencia McAllister, and she studies, pancreatic tumors. And she said that the pancreas has its own microbiome, but the pancreatic tumors have their own microbiome. So, then you wonder, all right, so every tissue type in an organism probably has its own unique microbial constituent, its own little microbiome, because the cells there, have different surfaces and make different things and need different things. So this is just, again, just my speculation, but in the human body, I would bet that, every time the nature of the cells in any part of our digestive system, for instance, changes, the microbial attachment changes.
Yes, and this is also really important. I mean, these local or geographic differences in microbial ecology is obviously crucial. I mean, the way we currently look at and test the system by taking a stool sample is incredibly crude, and it's actually surprising how much science has been made out of this up to now. I'm absolutely convinced the next 5 years this will change dramatically with these capsules. They can sample in different locations and either send out the information or you collect the capsule at the end and just get, for example, microbial communities or metabolites from one distinct area in the gut. And I'm sure there's already experiments in mice where this has been done. You can just take out the intestine and analyze in different regions. Where it's been shown that there's very different communities that produce very different molecules in all these sites. And what you said is that, that a tumor, there's clearly this bidirectional communication between the host cells and the microbes. So the host cells and what they do, what they produce, secrete, can influence behavior of the microbes, and vice versa. That tumors could actually, significantly change in a particular region. This could be true about, for example, don't even have to go to pancreatic cancer, colon cancer, opening up even a diagnostic possibility in the future that you would, if you sampled your colon with one of these capsules and find that the community is altered in a particular region, this could be a precursor of colon cancer. I always like to use this example or this analogy of the soil microbiome. The soil microbiome is different in different locations depending on how, even though you think soil is something universal, but depending on the composition of the soil, how it's been treated, how much water it gets, each of these soils will develop their own microbial ecosystem, which then plays a big role, interacting with the plants that are grown in these areas. So this is not a static system that once established early on in life stays the same for the rest of your life. This is clearly something that is plastic and adaptive to various influences, and it shows a great variation in different locations in the GI tract.
Yeah, no, this is great. What specifically are you working on right now?
So we have gone through a phase of we call extensive comprehensive phenotyping of people, healthy people, and people with different diseases or dysregulations of health, and which is essentially, in our opinion, this was the first step that had to be done to really say in humans there is a relationship between microbes and for example, pain sensitivity or a diagnosis like depression or irritable bowel syndrome. So we're just ramping up this phase of cross-sectional correlations of microbial communities, their metabolites, the metatranscriptome with human traits. And the human traits, we use brain imaging, battery of questionnaires, diagnostic questionnaires. So now the whole field is rapidly moving into the next step, which is longitudinal studies which are aimed to prove causality. And that's still a big question mark, in mouse models. So when you look at all the publications on mouse models, elegant studies, very mechanistic, and they provide pretty strong evidence that the microbes play a causative role in like emotional behavior or feeding behavior or but that's not the case in humans. So in humans, we're still, stuck with these cross-sectional associations. We cannot say, do microbes or change in microbes play a role in human depression or in human anxiety or autism spectrum? It's all associations in humans. So we are focused on doing this now in human studies, these longitudinal studies with interventions where you manipulate the gut microbial composition. Obviously, we're limited in humans. We can't do the same thing as in mice, but we can give antibiotics. We can do dietary interventions. And then hopefully, in the next few years, and these are expensive studies, much slower than what you can do in mouse models. Hopefully, we'll get an indication about causality in humans, what these microbes do. I mean, they clearly, they must have an effect, because otherwise you wouldn't see all these findings from the mouse models. On the other hand, the human brain is so much more complex than the mouse brain that it's conceivable that this influence of the microbes is much, much smaller on human behavior as it is on mouse behavior. So you have to look much closer to actually detect these causal effects. And if you look at the literature, I think you mentioned this in the beginning, there's a big discrepancy between very convincing mouse studies and human studies that would prove the same point in terms of causality. So this is our main interest right now to really provide this missing link from human studies. And we're looking at healthy people. We're looking at obese individuals with food addiction. We're looking at cognitive decline. We're looking at autism spectrum. So those are really the main areas. Many of these studies are done in collaboration, consortia, like the cognitive decline / Alzheimer studies being done part of a consortium with Duke. So in many ways, a very exciting area of research, certainly the most exciting part of my career. On the other hand, still a lot of frustration why we can't provide the definitive proof that this is important for human disease and human health.
Yeah, no, it is I consider it to be super exciting and interesting as well. Well, Emeran, unfortunately, we're out of time. I'd like to have you back sometime soon, but for now, I thought we brought up a lot of provocative, really interesting stuff. What's the best way for people to follow up and read some of the papers you put out? Take a look at the research you're doing right now. How can they get in contact?
So probably the simplest way is to go to my website, emeranmayer.com. There's a lot of information there from interviews, podcasts, also links to our publications and all the other social media channels. We also have a newsletter that we're sending out where you can sign up on the website for that newsletter. I would say that's and people that want to read, so the real background on this from an easily readable publication, the book I published a couple of years ago, The Mind-Gut Connection, is probably a good start for people that are not familiar with the field. I should mention I'm working on the second book right now, which is sort of updating the first one and taking it to the next level of understanding.
Well, Emeran, very good. I appreciate you coming on the podcast. And like I said, it's been great.
Well, thank you for having me. It was fun talking to you.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.